Dear student, welcome to this session. I am Srimati Jayashri, working as an assistant professor at the Department of Microbiology in KTHM College, Nasik. Today, I am going to deal with the subject Basic Techniques in Microbiology of FIBSC class, in which the topic is related to the staining techniques, and the lecture is delivered on the stains and types of staining techniques. As we know that the microorganisms present at everywhere, these microorganisms are very small, transparent and invisible to our naked eyes. So it is very difficult to observe these microorganisms. These microorganisms are only identified by the microscopic examinations. Microbial cells are colorless, so these cells are suspended in the aqueous medium. The refractive index of the cell and the aqueous medium is less or same, and due to lack of the contrast, the observation of such unstained microorganisms are very difficult. So, staining is a very essential step for the bacteriological studies. In today's session, we are going to learn about the dye modern stain and its component, then the types of stains, then classification of staining techniques, and in which we are going to see the simple staining and differential staining methods. After attending this session, students will able to know the concept of dye, mordant, stains and its type. Then they are classify the staining techniques. Then the students will understand the monochrome staining procedure as well as the gram staining technique. Let's begin with the concept of dye and stains. So dye is a coloring agent that is used for the general purpose, while the stain is that dye which is used for the biological purpose. So dye is organic compound used for the coloring non-biological agents or inanimate objects, and it is a single chemical reagent containing the stain. While the stains are the coloring agent which are used for the biological purpose, like staining of cells, bacteria and other microorganisms. So these stains are basically the mixture of selected dyes which gives the color to particular biological specimen. Now turning to the concept of mordant. Mordant is a chemical compound which is bring about the staining reaction. So these mordant are help to fix the color to the cell components. So these mordants are basically divided into two types, acidic mordant and basic mordants. So basic mordants are reacted with the acidic stains while the acidic mordants are reacted with the basic stains. For example, in Zill-Nielsen staining technique, phenol is used in the carbolfoxine, and in gram staining procedure, iodine is used as a mordant which forms the crystal violet iodine complex. Now turning to the purpose of staining. Staining procedure help in the production of contrast between microorganisms and in aqueous medium. So it is very useful to study the morphology of the bacteria. So morphology includes size, shape and different types of arrangement of the bacteria. While this staining differentiate one organism from the another. For example, in gram staining, the bacteria are differentiated into the gram positive and gram negative bacteria. This staining determines to particular structure of spores or the flagella. Now, the stainings help in studying the internal as well as external characters of the bacteria. Let us see the components of stains. Basically, stains are composed of the three components: benzene ring, chromophore group, and oxochrome group. Benzene ring is organic colorless solvent, while the chromophore group is colored bearing compound, and the oxochrome group is the ionization having the ionization property which acts as a color intensifier or color enhancer. Benzene plus chromophore produces the chromogen compound, and this chromogen is nothing but the colored compound only. Oxochrome with chromogen allows the dye to form salt component that adheres to the cells or that attach to the cell. Now turning to the classification of the stains. Classification of the stains which are based on the charges, they are classified into three types: acidic stain, basic stains, and neutral stain. Acidic stains carries the negative charge on it. For example, nigrosin, eosin, malachite green, India ink, and carbolfuchsin. While the basic stains carries the positive charge. For example, crystal violet, methylene blue, safranin, etc. And the neutral stain which carries the both positive and negative charge on it. For example, Giemsa stain, Leishman stain, etc. Let's study about the acidic stains. Acidic stains works best in the acidic pH and they carries negative charge and they are anionic in nature. These acidic stains have the affinity for the positive component of the cells. Acidic stains do not stain the bacterial cells, they stain the background only with the contrasting colors because the most of the bacterial cell containing the negative charge on it, while the acidic stains are also having the negative charge and which repels the stains and the bacterial components. That's why it's only color to the background, not to the bacterial cells. For example, eosin, rose bengal, acid fuchsins, India ink, nigrosin, and picric acid are some examples of the acidic stains. Now, the basic stains are works based in the basic pH. These are having the cationic in nature and having the positive charge. They have high affinity with the negative charge molecules such as the nucleic acid, proteins, and surface of the bacterial cells. So these basic stains can stain the bacterial cell. They possesses negative charge on it. That's why the basic stain is having the positive charge and the bacteria is having the negative charge and it attracts the bacterial cell. The stain is attracted to the bacterial cell and bacterial cell get stained. So for example, that is a methylene blue, basic fuchsin, crystal violet, safranin, and malachite green. Now turning to the neutral stains. Stains carries both positive and negative charge on it. For example, the Leishman stains. Mostly, this Leishman stain is used to stain the blood smear staining technique. In this, eosin is acts as an acidic stain and methylene blue is acts as a basic stains. So most of the bacteria is having the acidic components and negative charge. So they have high affinity with the basic stains. For example, methylene blue, and the bacterial cells appears deep blue color on the Leishman stains. Now the hematoxylin and carmine are the other examples of the neutral stains. Now turning to the classification of staining technique. Staining techniques are divided into the two parts, actually based on the stain used in the technique. If single stain is used, then the staining technique is divided into the direct and indirect technique. So direct technique is also called as positive staining, while the indirect technique is also called as a negative staining. In the positive staining, only one type of dye or stain is used, for example, the monochrome staining. While In the second type, we using the two types of contrasting stains. It divided into two types: differential staining technique and selective staining technique. In the differential staining technique, it differentiates the bacteria into two groups, and in selective staining technique, which is used for visualization of the structure of the bacterial cells. In differential staining, the bacteria are differentiated into two groups. For example, gram staining, in which the bacteria are differentiated into the gram positive and gram negative bacteria. And second example is the acid fast staining, in which the bacteria are differentiated into the acid fast bacteria and non acid fast bacteria. While in selective staining technique, which is generally used for the visualization of structure of bacteria, like the cell wall staining, PHB staining, capsule staining, flagella staining, and endospore staining. Now turning to the simple staining technique. In the simple staining method, only one type of stain is used, and which is usually sufficient to reveal the morphological features like the shape, size, and arrangement of the bacterial group of the cells. It does not differentiate between the different types of bacteria. Simple stains are generally used to stain the whole cell or the specific cellular component. It is also called as the direct stain or positive staining. So, in this direct staining or in positive staining, it will stain only the object, while in indirect staining or in negative staining, it stains the background only and it do not stain the bacterial cell. Let us see here, the bacterial cell is having the negative charge on it because most of the bacteria consist of the nucleic acid, proteins, and the bacterial cell wall is having the negative charge on it. When these bacterial cells are stained with the basic stains, which are generally positively charged, and it get attracts to the bacterial cell and the bacterial cell gets stained, or bacterial cell appears color because of the positive negative attraction of the bacterial cell and the basic stain. So this is generally observed in the direct staining procedure. While in indirect staining procedure, the bacterial cell is having the negative charge and it is reacted with the acidic stains, which are generally carries the negative charge on it. Then this negative charge is repels to each other, and the bacterial cell does not stain, only the background is stained in this indirect staining procedure. Now turning to the slide preparation technique. How will you prepare the slide for the staining? So first we have to take the clean and grease-free slide, then label the slide. After that, we have to make a smear on it. So you can make a smear from bacterial culture suspension, or we can use the bacterial colony with the water and spread the colony on the slide and make a thin smear on it. After that, air dye the smear and heat fix the smear by passing through the slide 2 to 3 times through the Bunsen burner flame. This heat fixation kills the microorganisms, then it helps to adhere the microorganism to the slide, and it gives readiness to the bacterial cell to accept the dye. So some problems are related to the smear preparations, like the smear is too thick. If the smear is too thick, the microscopic observation is very difficult. Then sometimes smear is too thin and it easily get washed off. Now turning to the monochrome staining procedure, which is the technique involved in a simple staining. In the monochrome staining procedure, only one reagent is used, in which make a smear and then dip these slides or soak the smear with 30 to 60 seconds in the stain, which is generally used the methylene blue. Then after treatment with stain, rinse the slide with water. Then we will observe the bacterial stains appear by the microorganisms. By observing these microorganisms under the microscope, we will uh, have the results like the purple color bacteria are observed under the microscope. So, in the simple staining procedure, generally the basic dyes are used, which are having the chromogen that is positively charged and the cell wall which is having the negative charge. This monochrome staining shows the morphology, that is the size, shape, and arrangement of the bacteria. So, the stains which are used generally in monochrome stainings are methylene blue, carbolfuchsin, crystal violet, and safranin. Now, in the simple staining, the second procedure is the negative staining method, in which the acidic dyes, which are having the negative charge on it, are used and which is repelled by the negatively charged bacterial cells. So, the cells remain colorless and seen against the dark background, like in the picture. So, we can see here the dark background versus the colorless cells. Now turning to the differential staining technique, in which two or more reagents are used. This distinguish basically the bacterial groups and specific structures. For example, the gram staining, in which the bacteria are differentiated into gram positive and gram negative bacteria, while in acid fast staining, the bacteria are differentiated into acid fast and non acid fast bacteria. Now turning to the gram staining procedure. Before turning to the gram staining technique, we should know about the cell wall structure of gram positive and gram negative bacteria. In gram positive bacteria, the peptidoglycan layer is very thick, while in gram negative bacteria, the peptidoglycan layer is very thin. This peptidoglycan layer is made up of the proteins and sugar molecules. This peptidoglycan layer is very thick in gram positive bacteria, while in gram negative bacteria, this peptidoglycan layer is very thin and which also consists of outer membrane, thick outer membrane, and having the perisplasmic space in the gram negative bacteria. Now, the which reagents are used in the gram staining procedures? First, the crystal violet is used for the staining purpose and which is acts as a primary stain, while the iodine is used in this staining technique, which acts as a mordant, and the decolorizer is made of mixture of acetone and alcohol, which is about the 95%, and the last, that is the counter stain, that is safranin, is used to stain the gram negative bacteria. Now turning to the principle of gram staining procedure. When we prepared the slide for the gram staining procedure, first add the crystal violet dye for 60 seconds. By this adding the crystal violet, as we know that this crystal violet is a basic dye and which is having the positive charge, that's why the negatively charged bacteria which are having the negative charge on it, which attracts this crystal violet towards the bacterial cells. But in gram positive bacteria, the thick peptidoglycan layer is present and the crystal violet is attached to that peptidoglycan layer, while by adding the iodine on the slide for the 60 seconds, the crystal violet and iodine complex is formed. Here the iodine acts as a mordant and which fix the color to the gram-positive bacteria. After that, the decolorizer is used. By this decolorizer, the gram-positive bacteria get shrink and adhere the crystal violet iodine complex, and some cells are decolorized by the action of decolorizer. They will get off the stains. Then after adding the counter stain, the colorless cells which are get the safranin or get the red color because these are the gram negative bacteria. So these gram negative bacteria, in which the gram negative bacteria, the peptidoglycan layer is very thin and it lose the CVI complex and the counter stain get color to these cells. That's why the gram positive cells are violet in color while the gram negative cells appears pink in color. So these are the result of the gram staining procedures. So gram positive bacteria color the violet color and the gram negative bacteria gets the pink color. In today's session, we have seen the concept of dye and stain. So dye are the chemical compounds which are generally used for the general purpose or generally used for the inanimate objects, while the stains are used for staining the bacterial cells or biological objects. Now the types of stains like the acidic stains which are having the negative charge on it, then the basic stains which are having the positive charge on it, and the neutral stain possesses the positive and negative charge on it. The staining techniques like the simple staining and differential staining. In simple staining, we have learned about the monochrome staining in which the single type of stain is used, while in the differential staining, the more than two stains or two stains are used. So in the differential staining, gram staining and acid fast staining is done. In the gram staining procedure, it differentiates the gram positive bacteria and gram negative bacteria. In acid fast staining, it differentiates the acid fast and non-acid fast bacteria, while in monochrome staining, the bacterial cells get colored while the background is contrasting, and in negative staining procedure, the background is colored and bacterial cell remains colorless.